Well, I finished doing the dishes, regrouting the bathtub, and cleaning out the stables of the 12 incontinent bulls. Tell me, O wise sword of righteousness, were these menial tasks meant to teach me humility?
No, they just needed to be done. I wasn't going to do it. Anyway.
While Earthworm Jim was tasked with something very trivial, this podcast will show that his Earthworm buddies were tasked with something far more important than he ever was.
Dendrobina veneta, commonly known as the compost earthworm or European nightcrawler, is a part of kingdom Animalia, phylum Annelida, class Oligochata, order Opisthophora, family Lumbricidae, and genus Dendrobina. This earthworm has made its way through ecosystems across much of the northern hemisphere, including the United States. The species was first discovered in the late 1800s. However, it has also been speculated that the species may have arrived during the early colonization of the Americas in the 1600s. The epithet veneta in the species name refers to the Veneto region in northern Italy. Because of this, they are often referred to as European nightcrawlers and closely associated with the Encina hortensis, another common composting earthworm. These earthworms are soil dwellers and can tolerate water, environmental, and moisture variations when compared to other species. Earthworms are triple blastic protostem coelomates. They have both male and female reproductive parts, have a closed circulatory system, and complete digestive system. Their life cycle can be completed in 100 to 150 days, and it only takes them 65 to reach sexual maturity. They can live from 1 year up to 5 years based on the environment. They often die from changes in the soil, drying, or flooding. Earthworms can be easily bred artificially in standard conditions without needing any additional materials and nutrients other than normal organic soil and veggies or oatmeal as food.
The Veneta are highly sought after by gardeners, composters, and fishermen alike due to their size, liveliness, and ability to thrive in various conditions. They are extremely useful in vermicomposting, a process that relies on earthworms and microorganisms to help stabilize active organic materials like scraps, paper, or leaves and convert them into valuable soil and plant nutrients. As the worms consume the matter, their digestive process turns the waste into worm castings or vermicompost, a nutrient-rich substance that helps other species thrive. Worm castings do such a great job helping plants thrive that they are valuable soil amendments used by organic farmers, vineyards, greenhouses, and more. However, it is important to note that outside of controlled systems, the species is invasive and the high tolerance to ranges in environmental parameters can be to blame. This leads to harmful effects on soil ecosystems. Most importantly, earthworms are valuable model organisms for laboratory experiments for many reasons. First of all, they have a relatively simple anatomy and physiology compared to mammals, often making them easier to study and manipulate in experimental settings. They possess remarkable abilities to heal themselves and regenerate lost tissues or limbs, making them very valuable in studying wound healing treatments such as PBM. Furthermore, they share many biological processes and genes with humans. They're easy to care for in a lab because they require such minimal space and resources, rapid generational turnover, and quick results. Also, they are highly sensitive to changes in their environment, including temperature, pollution, toxins, acidity, and moisture. Lastly, they easily meet the 3 R principles and National Science Foundation statement. They raise fewer ethical considerations, questions, and concerns for more complex species.
Due to their advantageous traits as a model organism, earthworms are great candidates for self-healing research and photobiomodulation. Photobiomodulation, or PBM, is a laser technique which uses low-intensity, non-ablative light sources to manipulate cellular activities in some mammals. It can be used on external surfaces of the body to trigger a cascade of metabolic effects and physiological changes, resulting in improved tissue repair, which can be relevant to improved treatments of superficial or deep tissue injuries, as well as degenerative and autoimmune diseases or aesthetic purposes. There have been a huge increase in clinical instrument technology and applications, creating a demand for reliable experimental models to test effectiveness. Because earthworms are so easy to use, already have some regenerative properties, and have been previously shown to be positively affected by PBM, they were chosen in a 2019 research study in the European Journal of Histochemistry to test if low-intensity light can help cells heal wounds.
Researchers hypothesize that if tested with PBM, Neonatal will be positively affected in accordance with previous cell experiments and demonstrate itself to be a reliable experimental organism which meets the 3 R principles in animal research. To test this hypothesis, an experiment was set up with Neonatal to evaluate the effectiveness of PBM. The effects of laser therapy on the infrared spectrum was examined on the wound healing of earthworms by performing 3 treatments every 24 hours. Each earthworm would be anesthetized on an ice pack for a few minutes before cutting 30 posterior segments. Using a dental laser, the worms were irradiated immediately after the excision, 24 hours after and 48 hours thereafter. Control worms were not at any point. Both treatments were kept in individual sterile dishes in organic soil and added water for up to 12 days for observation.
Effective outcome was assimilated by examining the macroscope, histological, and molecular changes of the irradiated segment of the excised worms with respect to controls. Results that showed that 6 days after the excision, 100% of both irradiated and non-irradiated segments were alive, with the irradiated segments appearing more vital and dynamic. After 9 days, 100% of the irradiated samples were healthy, while only 75% of the controls appeared vital, dropping to 50% after 12 days. By 16 days after the excisions, all the non-irradiated segments succumbed or died, whereas 100% of the laser-treated samples survived, with a very high significance, or p-value, of less than 0.001. After the evaluation of several other factors, the treatments were concluded as sufficient to promote wound healing due to the effects on the muscular and blood vessel contraction. The treatment of bacterial load, reduction of inflammatory processes, and tissue degeneration. The Veneta was demonstrated to be a reliable experimental organism that thoroughly meets the 3 R principles of animal research and the National Science Foundation statement. This is highly applicable to modern medicine and innovative technologies, as the research shows the differences between how wounds heal normally and how they heal using photobiomodulation and laser techniques. Results indicated that PBM has self-healing effects on wounds in D. veneta, which is genetically very similar to humans. This means that it is likely possible the technique could have similar biomedical effects in humans and other animals. Furthermore, it provides evidence and support for earthworms as valuable experimental models for investigating various advancements in human health and disease, as well as exploring broader biological questions. Most importantly, it demonstrates earthworms to be a valuable model organism in laboratory experiments due to their regenerative abilities, relative simplicity, genetic similarity, ease of maintenance, and more. In conclusion, through their genetic and evolutionary peculiarity, comparable to those of scientifically accredited models, D. veneta allows the effect of laser therapies by multidisciplinary methods with relevance to many other areas of study.